So today we're going to talk a little bit more about the cell theory itself. So there's only three major components of this, but there are some things that kind of go along with it. So we're going to make sure that we hit those three major ideas and then some exceptions to cell theory, a little bit about like how cells grow. So let's get started. So the first major concept when we're talking about cells is that all living things are made of cells or composed of cells. Now remember that this is theory, and part of it is almost like a definition, right? So we're defining life by our understanding of cells. Cells are the basic unit of structure and function in living things. So basically, all living things have to exchange matter with the environment around them. Cells have to exchange matter with the world around them. Living things need gas like oxygen or carbon dioxide, and they have to remove waste. So do cells. Living things need energy. So do cells. So we can look at life on the scale of an individual, or we can look at life on the scale of a cell, and the basic needs are going to be similar. And then we have that cells come from pre-existing cells. So the early work here, that living things are composed of cells, we could talk about Schleiden and Schwann, we could mention Hook and Leeuwenhoek. But down here, when we start talking about cells come from pre-existing cells, we're really focusing here on the work of Virchow. But as everything, there are going to be some exceptions to cell theory. So here's where they are. And we're going to talk more about these in depth later. But the first one is viruses. They're not made of cells, so that's an exception to cell theory. Mitochondria and chloroplast are also exceptions to cell theory, even though that they can reproduce all by themselves. And they have a lot of the same components that cells, basic cells have. They have their own genetic material, so they have their own DNA, they have their own RNA, they have their own ribosomes, they have their own membranes, and they do a lot of things independently. We don't consider them to be cells in and of themselves because they are not independent. They are still within a cell and using that cell for materials. When we're thinking about the average size of a cell, we're looking at something pretty small. So an egg, like a frog egg here, is actually pretty little. It's just a few millimeters in size. A human egg, you could actually see at the very, very tip of a pin. They're very small. Most of the animal and plant cells are going to be here in the micrometer. So it's a thousandth of a millimeter. So they're pretty tiny, and that's why we would need a microscope to see them. The other thing we're looking at are these like random, like the sizes of cells, right? And so when you're looking at this little bitty cell versus a big cell, we have this surface area. So you've got the 6 sides, right? So you've got the surface area, in this case of 6 versus a surface area of 150. And then the volume, the number of boxes that it's composed of is just 1, whereas this one's a little bit bigger. So the bigger the surface area is compared to the volume, it's going to be helpful in the terms of cells. So here's what we mean by that. Cells don't get bigger when you get bigger. They divide. They make more cells. And they do that in part because the surface area to volume ratio actually really, really matters. The more surface area has, the easier it is for matter and energy to get to the center of it. So the cells will stay small so that things can get in and things can get out easily. A great analogy when we're thinking about this is thinking about boiling potatoes. When I'm boiling potatoes to make mashed potatoes for supper, I don't throw an entire potato in the hot water and hope it's ready in the next few minutes so I can prepare food for my kids who are crying because they're hungry. I cut the potato up into tiny little pieces so the hot water can cook all of the potato faster because there's more surface area. So anytime we increase surface area, we're going to allow more things like heat to get into the cell faster to allow the processes, in this case cooking, to happen a little bit faster. And of course, we study cells with microscopes. Now, in the lab that we would be using if we were actually in the lab, right, would be a light microscope. So it's going to be the one on the left. We're using light. We can see, a couple hundred times bigger than what you can see with your eyes. The electron microscope is what this gentleman is using in a lab. They're huge. They're incredibly expensive, the cost of a house. And they allow us to see very, very closely in the cells. So a TEM is a transmission electron microscope. Now, it's not electronic. That means you plug it into the wall. Microscopes we have at school, you plug into the wall as well. They're all electronic now. But this transmission electron microscope lets you see inside the cells to the actual organelles. And we're going to look at some pictures of the inside of organelles later. The scanning electron microscope, SEM, is going to look at the outside of the cells, the cell membrane, the exterior, and give us a really close-up view. And we'll look at that a little bit more in future too. So I hope this helps. Please make sure you take notes and take note of any questions you have so that we can clear up any questions or misconceptions. Thanks for learning with us, guys. Talk to you soon.